Good morning to all. Today, we are going to discuss the Operon concept. And all of you know that the Operon, it is a set of related genes placed under the control of a common regulatory sequence. And cystron or a polycystolic mRNA is present in case of prokaryotes, where all the genes which needs to be transcribed together for a common task were placed under the control of a common regulatory element, and it is transcribed in a single unit or as a single polycystonic mRNA. Then subsequently it is translated into its concerned proteins, and usually these operons were designed for performing a particular task or a specific task. For example, lactose utilization or tryptophan biosynthesis, something like that. So the cystron is the component of the mRNA or a portion of the mRNA that is corresponding to the production of a particular peptide. In case of polycystonic mRNA, as you know, it contains multiple cystrons or multiple regions corresponding to concerned polypeptides. So the many polypeptides can be simultaneously translated from the same polycystonic mRNA. So that is the concept. When we come to the different types of genes, an inducible gene is one whose expression increases in response to an inducer or activator. So an inducible gene can be induced by an activator or an inducer molecule. So its response or its expression increases in the presence of the inducer or the activator. And there is another category of the genes which is known as constitutive genes. The expression of some of these genes, that is constitutive, meaning that they are expressed at a reasonably constant rate and not known to subject to any regulation. These are often referred to as housekeeping genes. Usually, such housekeeping genes were always required at a basal rate. Especially they are the genes which is contributing to some structural components and also for some basic cellular activities which is always required. So that type of genes were constitutive genes. In the absence of constitutive genes or in case of an anomaly in the case of synthesis of such constitutive proteins, usually the cell will not survive because it is a base requirement for the existence of the organism. So they are known as housekeeping genes. Whereas the inducible genes were produced as a response to the inducer or activator molecules, otherwise it is not expressed. So it is produced in response to an environmental stimulus or some other stimulating factor. Here you can see there are regulatory proteins that is involved in such gene expression. There are two types of regulations. One is positive regulation and the second one is negative regulation. In positive regulation, and here is the common structure of a gene, prokaryotic gene here. Here is the promoter, then this is the operator, this is the gene which needs to be transcribed. So the same structure is repeated in the same diagram. So an activator molecule can bind to the operator region in case of positive regulation. And once the activator is bound, it increases the transcription. That's why it's a positive regulation. So the activator or the inducer molecule which binds to the operator will increase the transcription activity. Then in positive regulation, without the activator molecule, there is no transcription or the gene is normally in a suppressed state or an inactive state. In case of eukaryotes, the gene is normally in an inactive state. So it needs to be activated by the activator molecule, then only it is transcribed. And in the second case, we have another system, a negative regulation system, especially that is dominant in case of prokaryotes, where the gene is normally active always. Even without any stimulation factor, the prokaryotic genes were normally ready to be transcribed, and that means gene is normally present in an active state in case of prokaryote. Whereas in the normal state of the eukaryotic system, it is present in an inactive state because, as you know, the eukaryotic genes were normally highly compacted or highly packed by binding or wound up by binding around these histone proteins, and the DNA is not made available for the transcription machinery to operate. So that needs to be activated and it needs to be unwound, and that DNA sequences need to be provided for transcription by interaction with activators and different other and protein complexes that is interacting at the operator level. Okay, so normal behavior of a eukaryotic gene is a suppressed state or an inactive state, which needs to be activated by unwinding and making the DNA available for transcription machinery to work on. So that is a positive regulation, especially that is dominant in case of eukaryotic system. Whereas in a prokaryotic system, as you know, there is no histone proteins. The DNA is actually present as a loose chromatin network, which is readily available for all the transcription assembly to bind to the promoter and operator regions and start the transcription. So without a repressor molecule, the prokaryotic genes were always active and transcription can occur. So for the regulation of a prokaryotic gene, especially it doesn't mean that positive is present in eukaryotes and negative is present in prokaryotes. Both the systems were present in both systems, but predominantly positive regulation can be expected in eukaryotic system, or it is required in eukaryotic system because normally the genes were inactive. And in prokaryotic system, normally the genes were highly active. It needs to be repressed for regulation. It needs to be repressed by binding of the repressor protein to the operator region to block the transcription. So prokaryotes normally regulated by binding of the normally active genes were blocked by or regulated by the repressor molecules. And in the normally inactive eukaryotic gene needs to be activated by the activator molecules to start the transcription. So this is the positive and negative, or the overview of the positive and negative regulatory mechanisms in prokaryotes and eukaryotes. Here is another illustration in which we have a regulatory protein or activator molecule which is shown here. So here we have an allosteric site for binding of an effector molecule, and there is a DNA binding site, and normally the DNA binding site cannot be occupied by the DNA, or it is an inactive state or allosteric site. This regulatory protein is normally inactive. But when an effector molecule binds to the allosteric site, it results in a changed conformation of the activator molecule, which enables that molecule to bind to the DNA. For example, a hormone will bind to the hormone receptor, which will result in binding to this regulatory protein and result in a conformational change in the regulatory protein that enhances the binding of that regulatory protein to the DNA, and that change or that binding activates the transcription of a eukaryotic gene. So an effector molecule is normally required for activation of the activator and subsequent binding and transcription initiation. Whereas in case of a repressor system or repressor molecule is normally bound to the prokaryotic genes or the target DNA, and when the effector molecule binds to the repressor, it will be relieved from the DNA and then it starts the transcription. Now when we come to the lac operon, is a classical example of the operon. Whenever we are discussing the operon concept, we have to explain the lac operon. Lac operon is having three different genes, LAC-Z, LAC-Y and LAC-E. These are the three genes which is present in the lac operon, which needs to be simultaneously transcribed as a polycystonic mRNA and translated. And now there is an operator region immediately upstream, then there is a promoter site, then there is a LAC-I gene which codes for the LAC repressor. So LAC-I promoter, operator, like Z, like Y, and like A. These are the different components or different portions of the LAC operon. Okay, this is another illustration. The LAC-I gene, which codes for the mRNA, polypeptide, LAC repressor protein is produced. The LAC repressor protein will bind to the operator region. This is the promoter where the RNA polymerase binds, and if the LAC-I gene product, that is the repressor, is bound at the operator region, it blocks the RNA polymerase movement from the promoter forward. So, which effectively blocks the transcription of the structural gene Z, A, and Y. Okay. Now, in the medium, there is lactose. If there is lactose present, normally the lactose is not lactose that is acting as the inducer in case of lac-operon. As you know, it is allolactose, which is an isomer of lactose. And why this allolactose is used instead of lactose? That can be that's an interesting fact, because the system should not trigger the transcription of these structural genes by the presence of a few molecules of lactose. It should ensure that there is sufficiently larger quantity of the lactose present in the medium so that the operon starts translation and production of the concerned structural genes. How it is ensured? Lactose and allolactose were isomeric forms, and the allolactose is a rare isomer. For example, if 99 molecules of the lactose is present, then allolactose is only 1%. So the presence of one molecule of the allolactose indicates the presence of 99 molecules of the lactose. So the lactose as such cannot trigger the transcription by, or it cannot act as the inducer. The rare molecule that is a isomer of these lactose, that's allolactose, will bind to the repressor protein so that it can relieve the repression. So which means allolactose, if it is present, then that indicates the presence of a very large quantity of the lactose present in the medium, because it is a rare isomer. For example, 99 is to 1% or proportion. In that proportion, that lactose, allolactose concentration will be there. So in the absence of lactose, there will not be any allolactose. In sufficiently larger quantities of the lactose, allolactose will be present in very small quantities, and that can bind to the repressor molecules, and then it relieves the repression. And once the repression is relieved, then the RNA polymerase can move forward, and it can produce these polycystonic mRNA, which contains the structural genes Z, Y, and A. That will subsequently, that will code for beta galactosidase, then galactoside permease, and trans acetylase enzymes. Okay, and this part you I think you already know these things. So now when we discuss these regulatory mechanisms, here you can see there is a high glucose level. At high glucose level, you can see that there is ATP, and it inactivates adenylate cyclase. So the adenylate cyclase enzyme is actually is inactivated. So cyclic AMP is not produced. At a low glucose level, no inactivation, so the ATP to cAMP conversion is occurring at a low glucose level. So the presence of cyclic AMP indicates low glucose level. Absence of cyclic AMP indicates high glucose level. So this is the basic concept of the positive regulation mechanism in case of lac-operon, or activation mechanism in case of lac-operon. If cyclic AMP is present, it ensures that low glucose is present in the medium. In the presence of high glucose, cyclic AMP cannot be produced. So if there is cyclic AMP, that will bind with the catabolite gene activator protein or CAP protein to form a complex. Cyclic AMP cap complex will be formed, and this complex can bind to the promoter region so that it facilitates the polymerase binding. So the positive regulation in case of lac operon is through the mediation of the cap cyclic AMP complex binding to the promoter region, and that enhances the polymerase binding and positive interaction or the enhancement of the transcription activity of the lac operon. So this positive regulation system ensures that there is low glucose or there is no glucose present in the medium. At high glucose level, cyclic AMP will not be there. This cyclic AMP cap complex cannot bind to the cap site. So transcription or the RNA polymerase binding and subsequent translation cannot or transcription cannot occur in the system. So here, this is the lac I gene, which is coding for the concerned mRNA. Repressor proteins are produced, and the tetramer of the repressor protein will go and bind to the operator region, effectively blocking the transcription. The RNA polymerase actually it can bind to the promoter region, but that binding will not be strong in the absence of the cyclic AMP cap complex, which is a positive regulation element. So RNA polymerase cannot transcribe operator or distal genes ZYA cannot be transcribed because of this block and because of these feeble binding of the RNA polymerase in this system. Okay. So this is a no inducer or inducer and glucose. In both cases, even this transcription cannot occur. Then with inducer and no glucose, what happens is that the repressor molecule will be bound with the inducer, that is allolactose molecule, and the repression from the operator is relieved or it is released. And since there is no glucose, cyclic AMP cap complex can bind, and effectively it increases the binding of the RNA polymerase, and the RNA polymerase can move forward. In the absence of the repressor molecule, it can move forward and start the transcription of the genes Z, Y, and A. So that polycystonic mRNA is produced, and the concerned proteins, that is a beta-glucosidase and permease and transacetylase, can be transcribed from these mRNA. Now when we come to the details of this translation regulation, this is the same structure that is shown, that is a lac-I cap binding site or in the promoter region, promoter, operator, and the three structural genes. Okay. Now we have to understand 4 different conditions for, in lac operon. So if you understand the 4 different conditions and the system, how it is actually well regulated, it will give you a better understanding on the regulatory mechanisms of the positive and negative regulation. You take the example of low glucose and lactose available, high lactose. So lactose available and low glucose. That condition can be considered. So since there is low glucose, what happens? Since there is low glucose, cyclic AMP is there, cyclic AMP cap complex can bind, which promotes the RNA polymerase binding. Since there is lactose available, it relieves the repression, and repressor molecule will not be there in the presence of lactose isomer, that is allolactose, and that repressor will not be there. So this activated cyclic AMP bound RNA polymerase can move forward, and the lac genes were strongly expressed. So this is the only condition when the gene is or the regulated gene lac operon is expressed, because it ensures that there is low glucose present through this positive regulation system, and there is lactose sufficiently available through the the negative or repressor mediated system. So both systems were simultaneously used for ensuring that there is no glucose and high amount of lactose is present in the medium. Now take another case where there is high glucose present and lactose unavailable. So in the presence of a high amount of glucose, there is no further need for degrading lactose or breaking down lactose to produce glucose. It is an unnecessary wastage of energy. And also, the second condition, you may note that there is no lactose present at all. So in the absence of lactose and in the presence of high amount of glucose, there is no reason for production of the concerned structural genes. So the system will recognize the high amount of glucose by the absence of cyclic AMP cap complex. So it is not there, RNA polymerase cannot bind. And the lactose is unavailable. So allolactose is not there and the repressor protein will stay on the operator region. So the lag genes were not expressed. The third case, low glucose and lactose unavailable. Both glucose and lactose are unavailable. Okay, in this case, since there is no glucose, cyclic AMP complex can bind because CAMP is there, cyclic AMP complex can bind to the promoter region. It may, in that case, maybe RNA polymerase, there is a possibility that RNA polymerase can bind. But since lactose is unavailable, repression cannot be relieved. Allolactose is not there, repression cannot be relieved. So that means even though the positive regulation mechanism works, the negative regulation mechanism, that is the repressor mediator regulation mechanism, blocks the transcription and RNA polymerase binding, so it cannot be transcribed. And the last case where high glucose and lactose, both high glucose and lactose. At high glucose, the cap binding site cannot be occupied by the CAMP because since there is high glucose, there is no CAMP. Cap complex cannot be bound with the CAMP's cap complex. So the promoter region cannot be occupied by the RNA polymerase. Then since there is lactose available, of course the repressor cannot may be relieved. But since in the absence of the positive activation mechanism, the RNA polymerase cannot bind and start transcription. So nothing will bind here. All the promoter and operator regions and cap binding sites will be empty in case of a high glucose and a high lactose. So if you can explain these four conditions, that is a high, no glucose, no lactose, high glucose, low lactose, then high lactose, no glucose, then low glucose and high lactose. All these different conditions can be simultaneously assessed for understanding these regulatory mechanisms of a positive regulatory mechanisms and negative regulation mechanism in case of lack of flow. So this is another picture in which the regulatory mutants of the lac is evaluated, and you can see that this is in some cases where such complementation studies were done in case of molecular biology. This is the LAC-I gene, this is the polymerase binding site, then LAC-Z and LAC-Y. So some mutants of this operon may result in different type of gene expression. For example, in the first case, we can have the repressor which is coded by the LAC operon, which will bind to the promoter, blocking the transcription. Okay. And in the second case, we have a LAC repressor which will interact with IPTG. And in the presence of this IPTG, or this repressor molecule can be, it is removed with the IPTG, and that repressor molecule, it will not bind to the promoter region, then transcription will occur, so the concerned proteins will be produced. And in some cases, there is a LAC-I mutant will be there. The LAC-I mutant cannot produce the repressor molecule, so it will be like a constitutive state will be there. It will always express in the absence of the repressor molecule, the operon will always code for lag Z and Y. And of course, A. A is not shown here. And here lag OC mutant in which the repressor is produced, but there is a mutated promoter region. So in the mutated promoter region, the repressor cannot bind. So the gene is in a constitutively expressed state. Regulation mechanism will not work in both these cases, like LAC-I mutant and promoter mutant. And in this case, E, you can see that the repressor is produced, and in two genes, in this one repressor is there, and the other gene there is no repressor because of the mutated LAC-I gene. So if both genes were present in the same system, the repressor can bind to both the genes and inactivate both genes. And we can expect expect such a case when, say, one cell or E. coli cell is having a mutated LAC-I gene, or which results in a disabled or dysfunctional operon regulation system. And we may introduce a vector, a viral vector, or a plasmid vector, which contains a new gene with LAC-I and other functional genes into the system. So the vector mediated gene product, that is LAC-I gene, will also bind to the promoter of the host cell promoter region, and it also binds to the plasmid promoter, and also it will bind to the host cell promoter region, effectively blocking both of them. So this is a part of complementation. So this gene, or the LAC-I gene of the incoming vector, will complement the regulation of these host cell regulatory mechanism. And there is another example. In this case, we have a mutated promoter region. So the LAC-I gene will it cannot bind here. It will be expressed, and in this case, since the promoter is mutated here, it will express because this regulatory element cannot bind here. But in other case, if there is a promoter, functional promoter, it will bind there, and this expression of this gene will be blocked. So a dysfunctional promoter or a mutated promoter will result in binding of in the transcription of these concerned structural genes. And here is another example in which, for example, a promoter is not there, so LAC-Z gene will be produced, and LAC-Y gene is mutated here, LAC-X gene is mutated here. So in this case, nothing will be coded for because it is repressed by the repressor molecule. But here, repressor is binding of the repressor is blocked, and LAC-Z gene can be coded, and LAC-Y is also blocked by mutation. So there are specific mutants of the LAC system, LAC-Z system, can be created, so that especially this is used as a tool in genetic engineering studies, and for screening and selection of the successful mutants, we need selectable and scoreable markers, and this LAC-Z system will act as a specific or designed selectable selection system for the presence or detection of the presence of such recombinant products in recombination or RDNA technology systems. So LAC-Z systems and its different types of mutants were generally used in genetic engineering for complementation studies and screening and selection of the successful mutants. The next one is tripoperon. As you know, tripoperon is an operon that is used for biosynthesis of tryptophan. So the biosynthesis of tryptophan is required when tryptophan levels were normally low in the medium. If sufficient quantity of the tryptophan is available in the medium, there is no need for production of or biosynthesis of this tryptophan. So the system should recognize that there is no tryptophan or there is a low quantity of this tryptophan is available in the medium, so that it requires biosynthesis using its structural genes, that is TRIP A, B, C, D and E genes. Okay, so this is the structure of the tryptophan operon. Here we have a promoter, operator, TRIP leader region or TRIP L, there is an attenuator site, then 5 genes, structural genes, which will code for the different enzymes which is used in the tryptophan biosynthesis. And this is the tryptophan molecules. This is the TRIP repressor gene, which codes for the mRNA and TRIP repressor protein. And this TRIP repressor will form the active repressor by binding with the tryptophan molecules. So 2 tryptophan molecules were required for the TRIP repressor to become active repressor, and that active repressor will bind to the operator region. And this is how the negative regulation works in the TRIP operon. And the second type of regulatory mechanism which is seen in case of TRIP operon is the attenuation based mechanism using the leader sequence, that is TRIP L sequence, and the attenuator sequence, which is shown here. So the attenuator system, if it is functional, then the attenuated mRNA will be of a shorter length. The structural genes will not be coded. And if both the systems were not working, that's repression system and attenuation system, if both were not working, then the TRIP or mRNA, all the mRNAs will be coded and the structural genes will be transcribed. Here you can see there's a scenario in which high level of tryptophan is present in the medium. So what happens? The complete leader peptide is produced. So this is the mRNA, this is the ribosome complex, and as you know, the leader region, in the leader region, there are 2 regions or two triplet codons which demands tryptophan. Okay, and these are the trip codons that is demanding the incorporation of 2 tryptophans to the new peptide. So here we have the 2 trip codons present in the mRNA, which demands the incorporation of 2 tryptophan molecules. Now this is the ribosome unit, and this is the region 1 of the mRNA. This is the region 2 of the mRNA, region 3 and region 4. So in TRIP operon we have 4 different regions, 1, 2, 3 and 4, followed by multiple U residues at the end near the 3' end of the mRNA. Okay, so these 3 and 4 regions, as you know, it forms a stem loop which results in the, or this is the stem loop which results in the termination of the transcription. Now, this is the DNA, this is the TRIP leader region which is shown here, and in the high concentration of tryptophan, both the TRIP operons will be the tryptophan residues will be delivered here, and the leader peptide will be completely synthesized. The ribosome will occupy the region 1 and 2, and the region 3 and 4 will form pairing in between them and results in the formation of a stem loop. And if the stem loop is produced, the RNA polymerase will be released and the transcription ends by production of a shorter mRNA like this one, attenuated mRNA. Okay. So in the case of a low level of tryptophan, you can see that the leader peptide synthesis will not be completed because of the shortage of two tryptophan molecules here. So in the scarcity of tryptophan, the polypeptide cannot be extended by incorporating the tryptophan molecules at this point. So the ribosome will halt at the region 1 of the leader peptide or the leader region. And that results in the formation of a pairing between or a loop between 2 and 3 regions of the leader region. And these 2 and 3 regions is an anti-stem loop. So once it is formed, then 3-4 pairing is not possible, and if 3-4 pairing is not done, then the stem loop cannot be created. So that results in the completion of transcription and the RNA polymerase of the completion of transcription of the leader region, and subsequent the RNA polymerase will proceed by transcribing the structural genes of the trip operon. Okay. So these are the key points. Bacterial genes are often found in operons, and genes in an operon are transcribed as a group and have a single promoter. Each operon contains regulatory sequences which act as binding sites for the regulatory proteins that promote or inhibit transcription. Regulatory proteins often bind to small molecules which can make the protein active or inactive by changing its ability to bind to the DNA. Some operons are inducible, meaning that they are turned on by the presence of a particular small molecule. Others are repressible, meaning that they are on by default but can be turned off by a small molecule. So the inducible molecules, also the inducible operons, can be induced or turned on by a small molecule inducer or activator, and the repressible genes can be turned off by a small molecule like a repressor molecule. The lac operon of the E. coli contains genes involved in lactose metabolism, and it's expressed only when lactose is present and glucose is absent. Two regulators turn the operon on and off in response to lactose and glucose levels. The lac repressor and catabolite activator protein CAP. So lac repressor is the negative system. Catabolite gene activator protein, that is CAP, is the positive system of regulation in case of lac operon. The lac repressor acts as a lactose sensor. It normally blocks transcription of the operon but stops acting as a repressor when lactose is present. The lac repressor senses lactose indirectly through its isomer allolactose. Catabolite activating protein, or CAP, acts as a glucose sensor and it activates transcription of the operon but only when glucose levels are low. CAP senses glucose indirectly through the hunger signal, that is a molecule of cyclic AMP, or in the absence of glucose, cyclic AMP will be present. So this cyclic AMP act as a hunger signal or a demand for the production of glucose molecules by breaking down lactose. The tripoperon found in E. coli bacteria is a group of genes that encode biosynthetic enzymes for the amino acid tryptophan. The tryptophan operon is expressed or turned on when tryptophan levels are low and repressed or turned off when they are high. The tripoperon is regulated by the trip repressor And when bound to the tryptophan, the trip repressor blocks the expression of the operon. Tryptophan biosynthesis is also regulated by attenuation mechanism, or a mechanism based on the coupling of the transcription and translation. So this is all about the Operon system, especially the LAC and the trip Operon based systems. And through these mechanisms, you can easily explain positive and negative regulation. And positive regulation, an example is the positive regulation of lactose operon through CAP by cyclic AMP CAP complex, which activates the gene and RNA polymerase binding. Then repressor mediator system that is acting on lacoperon, and of course it is also known as an inducible system using the inducer. In tripoperon we have a negative or a repressor mediator system, and also we have an attenuation based system. So these are all different types of regulation mechanisms in case of a operon.